Hello, welcome to your virtual epithelial lab. I am going to record this on WebEx, and I'm hovering down here on my shared screen. I'm going to walk you through all the steps that you need to be able to do this lab successfully. I make no guarantees. Multiple cats will not interrupt us. This one right here is Joy, but her name is Moo. Goes by Moo. She identifies as a brat. Other cats, I am sure, will make appearances anytime I'm talking out loud, whether on the phone or recording a video. The cats show up. All right, so let's get started. As we're going through this, keep in mind that I am going to go a little bit more quickly than I would if we were actually in lab together. And that's because you have the luxury of just pausing at any time. So if you are a little unsure and you need a little extra time to maybe read the instructions or catch up, just pause the video. You can rewind, you can rewatch, take your time. I'm not really going to be looking directly at you because I have a dual monitor here, and my dual monitor's got the split screens that's got the tabs that I need. The first place that I would start is with the Canvas page. I made a recording of the information that I would normally give to you before the start of lab, like at the beginning of lab. So normally we would convene and I would start by giving you some information about the material that we're going to be going over together during the lab period. I might show a couple of PowerPoints, draw some things on the board, that kind of information. So I recorded that, and that video is available here. By the time this video is available, the closed captioning should be done automatically by YouTube. And then this next step here is what I'm recording now. So the demonstrating how to use the University of Michigan's virtual histology microscope is what we're going through right now. So if you haven't already, go to this Canvas page and click on this link. When you click on this link, it'll bring you to a page that should look like this. What you need to do is click on this blue button. And the blue button is going to bring you to a Dropbox online storage page. And they ask you on this homepage here, like, please do not request to access the Dropbox folder. What they've done is they've made a file that you can download and save. And everything you need is linked to this file. So you just go to File, Save As, and actually, no, I'm gonna change the instructions on that. Instead of going to Save As, go to Download. Let me edit that here. Instead of Save As, we're gonna do Download. Go ahead and download this. And once you have it downloaded, you'll be able to click on the links that you'll need to do the histology activities. The next thing that you should do is just open your LT assignment. When you open your LT assignment, it should look like this. Go ahead and do all of the pages in order. Read through all the introductory material on this overview page. Read through the introductory material on the second page. Where you see blue words, those are hyperlinks. And if you click on them, they're going to give you more information. I had to get out of editing view, so let me go back there. Okay, So if you click on them, it's going to give you more information. Make sure you look through all those hyperlinks. They are important and read the information that they provide. You can start doing the activities on this second page. And then the histology activities actually begin on page 3. So I'm going to skip ahead to that. So the first thing we're going to do is we're going to look at an example of simple squamous epithelium. And what we're going to be looking at is simple squamous epithelium within an alveolus that is a structure that you can find in your lungs. So I want you to familiarize yourself with what that alveolus would look like in a histology slide. To give you a little bit further context, let me go into here. The University of Michigan tutorial has a little bit of background information. Here we go. This is found on page 156. So here they're showing you what the respiratory tree looks like. This is the trachea, otherwise known as the windpipe. It's going to bring air from your oral cavity down into your lungs. The trachea branches into these bronchus branches. This is the right bronchus branch because it's that individual's right. So as we're looking at that individual, this would be their right side and this would be their left side. So this is the right bronchus main stem. This is the left bronchus main stem. And then the bronchi branch into the bronchioles. And the first branch is called the primary bronchiole. Where it branches again, that's called the secondary bronchiole. And then where it branches a third time, this would be called the tertiary bronchiole. The tertiary bronchiole then has all these little circular structures on the end called alveoli. The single for that is alveolus. And these are the little air sacs that expand when you breathe in and contract when you breathe out. And these are the little air sacs that allow air exchange. So what we're going to be looking at today is histology tissue of a lung section that has been taken at the level that you should be able to find some tertiary bronchioles as well as a lot of alveoli. So to familiarize yourself with what an alveolus is going to look like in histology, I want you to open up your Connect Anatomy and Physiology Revealed tool. So to do that, log into your Connect platform that you purchased for your lecture materials. And I had it set up in advance, but of course it timed out. So let me log into that. I'm going to go to student view. I'm going to show you where to find this stuff in your particular view. So in your Connect platform, if you scroll down to resources, you'll find this link called Anatomy and Physiology Revealed, or APR for short. Click on that. I'm going to pull this over here. Okay. And then we're going to continue along with these instructions. So it says, look up what the structure of an alveolus looks like. Access the APR, log in to connect, click on APR, select module 11, respiratory. So up here under modules, grab this drop down, open it up, and come down here to number 11, respiratory, and select that. Next, it says click on the microscope icon in the top menu bar for histology. So here's this top menu bar, and here's the microscope icon. And when you select that, the topics that are available in the drop-down menu will all be histology related. It says select topic alveolus. So we're going to click select topic, and we're going to come down here to alveolus. For select view, let's start with high magnification. So this is what a cross section of an alveolus looks like. Remember that an alveolus is a sac, so it's almost like a balloon, like a three-dimensional balloon. But when people prepare histology slides, what they're doing is they take a very, very thin slice of that tissue. So it's like they're putting a plane through it. When you take a thin slice of a circle, what it gives you is something that looks like, when you take a thin slice of a sphere, like a balloon, what it gives you is a two-dimensional structure that looks like a circle. So that's what we're seeing here. We're seeing a thin slice of a sphere that appears like a circle. Now, a lot of the cells that make up this alveolus are the simple squamous epithelial cells we're interested in looking at. However, there are also some immune cells that are there as well. So let's take a look at what those immune cells look like. They're called alveolar macrophage. So if you click on alveolar macrophage, it'll highlight them. And the ones that got highlighted were these very, very round cells. Really, really round ones. The remaining cells that make up this alveolus are the simple squamous epithelium. And you might be able to see here that these cells are pretty flattened. They're definitely irregularly shaped. And there's only one layer of them. So those are our simple squamous epithelium. Let's look at the low magnification. At low magnification, you see what the lung tissue looks like, and you can appreciate that there's multiple structures in that lung tissue besides just the alveoli. The majority of these structures are alveoli. However, some of these thicker, kind of more pronounced and regularly shaped structures, these are actually bronchioles. These are probably those tertiary bronchioles that have branched multiple times off of the bronchus. So we're not interested in those. You definitely don't want to focus on those when you're doing your histology. You want to focus on these other structures that are the alveoli. So now that we have an idea of what we're going to be looking at, we're going to go back to that University of Michigan manual. And I want you to scroll to the bottom of page 159. There's my scroller. Okay, I'm going to go to 159. Here we go. Click on slide 130-1. Here's 130-1, lung 40x H&E. 40x is the objective that they use to magnify that slide. Remember that the ocular is also going to magnify tenfold. So this is going to be a 400x total magnification. We're going to click on this link and it's going to bring up this histology slide. To operate this, it's really, really simple. You can click and hold and you can drag this tissue around. Or you can use these pan keys down here. It's going to pan that tissue around. To zoom in, you can click on this plus sign, and it'll zoom in increments. Or you can use the slider, and it's going to zoom in based on how much you slide. Another way, am I not including audio? Sure hope I've been including audio. Now I'm going to be re-recording. Yay! Okay, so let's come back here. You can also single click to zoom in, and you can double click to zoom out. What I want you to do is kind of move around. I want you to look at the tissue at kind of a low magnification. What we see here is it's probably one of those bronchioles, probably a larger one, maybe a primary or a secondary one. And then these other, just happened. I didn't do that. My mouse kind of took over there for a moment. This is cat number 2. This is Balder. Some of these other structures like this are probably tertiary bronchioles. Let's zoom back out a little bit. This is probably a tertiary bronchiole. This is probably a tertiary bronchiole. And over here we have a blood vessel. If we zoom in and look at this a little bit more closely, you can tell that it's a blood vessel because it's full of all these cells. The majority of these cells are red blood cells. Of all the cells in your blood, red blood cells make up 99%. White blood cells make up 1% of your blood. So these kind of brownish, maybe bluish colored cells that you see, these are all white blood cells. They look much, much different because they have nuclei within them. And so they stain with that blue dye. The red blood cells don't have a nucleus because red blood cells are the only mature cell in your body that doesn't have a nucleus. And that's because they kick the nucleus out by exocytosing it so that they can specialize in just carrying oxygen around and they don't have to waste any space with a nucleus inside of it. So it's a really, really easy way to find blood vessels as you look for all these little pink cells that don't have any blue stain in them. Those are the red blood cells without a nucleus. The very few cells that do stain blue and are a little bit larger, those are the white blood cells. And if you look really closely, you'll see some little dark dots, and those are actually your platelets. Those are your blood platelets that help you with blood clotting. So kind of pay attention to recognizing blood vessels because we're going to be doing that today. Bye, Balder. And we're going to be looking at blood vessels in future labs as well. Let's zoom back out a little. I want you to find a nice, pretty alveolus, and I want you to find one that's near a capillary. So that would be a small vessel. This is a pretty good sized blood vessel. This one right here, this is a capillary. This is a small blood vessel. Let's take a look at that. Just like how we learned in week 1, if you want to zoom in on something, put whatever you're interested at looking at higher magnifications, put that in the center of your view, and then, why am I zooming out? Single click is zoom in. Oh, there we go. Put it in the center of your view, and then zoom in. Okay, so I like this alveolus right here. It's pretty good looking. I'm going to zoom in one more time. And I can't quite get that one in my view. So I'm going to use maybe this one instead. And then here's my capillary. At this point, once you've got an alveolus and a capillary in your view, you want to take a picture of it. Okay, so it says find an alveolus, use the slide bar or zoom in. Until a single alveolus fills about half your screen, make sure you include a capillary. Take a picture by using the print screen key or function on your keyboard. Alternatively, Windows users press the Windows key and start typing in snip. And what you'll see is that snipping tool will appear, and click enter. And my snip tool is open, but I'm not sure which window it's open on. Got a lot of windows here. Hi, buddy. All right, it's not open on either of those two. There it is. I'm gonna drag this here. I'm gonna put this guy Sometimes resizing windows is half the battle. Down here, and I'm gonna leave my Michigan manual here. Okay, so I got my snip tool open. I'm gonna click new, and it allows you to make a selection. So I'm just going to select the part that contains my alveolus and my capillary. And what that does is it brings up an image that you can save. So I'm going to save that. Just going to put it in my download folder. I'm going to save it over this old one. And that is all you need to do to complete step 5. If you use a Mac, you may use Command Shift 4, and that will allow you to highlight and select your area of interest. Once you've saved that image, you're going to upload it. So to do that, you're going to come back to your CuraCloud lab activity here and click on Choose Image. And I need to go to my Downloads folder, and I'm going to click on Alveolus and open it. And that is going to load the picture that you just saved into your CuraCloud activity. Now we can annotate it. Click here on this blue selector called Annotate. And it's going to bring up some annotation tools on the left. If you're zoomed in too much, the annotation tools might be collapsed. So you may want to adjust the zoom on that window browser by holding the control key and hitting the plus or the minus key until you can see these annotation functions. Now, for this part, I'm going to pull this over here so that we can continue to follow. Oh dear, too many windows. I want to continue following our instructions in LT. I got to figure out where that went. I don't remember closing that window. I'm going to get rid of this one. Let me just open it up again. Okay, so we're going to continue with these instructions. Upload a Cura Cloud. I just did that. And use the annotation function to label the following alveolus. So you can click on this pencil tool and it will allow you to draw around a structure. So I'm going to draw around that structure. I'm going to draw around my alveolus. And then I want to label that. So I'm going to click here on this text tool right here. And then click where I want my label to go. And I'm going to write alveolus. Alveolus is singular. Alveoli is plural. Now I need to find an alveolar macrophage. And if you remember, those were like the really, really, really round ones. So this one right here looks really round. So does that one right there. So I'm going to take my annotation tool and I'm going to switch colors just so that we can kind of be able to differentiate easily. And I'm going to circle that little alveolar macrophage. I'm going to go back to my text tool, select that, come back here and write alveolar macrophage. It's okay if you want to abbreviate things. If you don't like the position of that label, go to your select tool, click on that, and then you can click on your label. You can move it around. Remember, if I'm going too fast for you, pause the video, catch up. Now, the majority of our alveolus is made up of our simple squamous epithelium. So I'm just going to point out one of those. I'm going to select this arrow tool, and I'm going to find one that looks nice and flat. And I think this one looks pretty flat, but I'm going to change the color here. Let's make this blue. Okay, so I'm going to click on my selection tool. I'm going to click that arrow and select it. I'm going to change it to blue. And now I'm going to come back here to my text tool and I'm going to say that is a simple Unfortunately, you can't do multiple lines. So I'm going to click this text tool again. I'm going to make another text box, squamous epithelial cell. If you need to, it's okay to abbreviate. And lastly, I am going to, let's use green for the capillary. I'm going to point out my capillary. Click on the text, type in capillary. Okay, now when you're done labeling everything, make sure that you've done all the steps. So I've got my alveolus, that's in black. I've got my alveolar macrophage, it's a nice round cell within the alveolus. I've got my simple squamous epithelial cell, and I've got my capillary. Once all of those are uh, labeled properly, you can click this checkbox, and it's going to save all of the annotations that you made to CureCloud. And when you're ready, we can move on to our second histology image. We're going to move ahead to page 4. Make sure you complete all the other questions on your own time, but we're just going to focus on the histology ones together. So I'm going to move to page 4. Now we're going to be looking at cuboidal epithelium. We're going to look at an example of simple cuboidal epithelium. It says open holes A and P. So open your textbook. I'm going to come back here. I'll open my textbook. Let's go to figure 20.10 and 20.11a to see what renal tubules look like. Okay, renal tubules, proximal and distal, and collecting ducts look like. So figure 20.10. Oh, I'm in chapter 19. Let me move to chapter 20. Urinary. So this is page 254, it looks like. Okay, so we come down here. Here is figure 20.10. And these are some of the structures inside the kidneys. You don't need to worry about what they do. You'll learn about that in anatomy too. We're more concerned with looking at the different structures that you can find inside the kidney. If you come down here to 20.10b, you can see that near to this structure called the glomerulus, we have tubes that are called the proximal tubules. And that's because these tubes are near the glomerulus. And then far away from the glomerulus, we have some tubes called the distal tubules. And those tubes then eventually connect to collecting ducts. The proximal and the distal tubules and the collecting ducts are all made out of our simple cuboidal epithelium. Remember, simple epithelium is specialized in absorption, filtration, secretion, and excretion. So the simple epithelium and the glomerulus does a lot of filtration, and the simple epithelium in the proximal and the distal tubules does a lot of absorption and excretion. So we want to find some tubules. So it says in the University of Michigan manual, go to page 205 and click on slide 204, kidney H and E. So I'm going to go back. And we are going to move to page 205. Aha, there it is. And select slide 2 for kidney H&E. Okay. So when you select that slide, it should look something like this. This is a thin section of kidney tissue. And this inner part of the kidney is called the medulla. Anytime you have like an inner region and an outer region, the inner region is usually called the medulla, and the outer region is called the cortex. Those little round structures that we saw that were called glomerulus, you find a lot of those in this outer cortical region. But we're interested in finding tubes. So we're going to stick to this kind of inner medullary is the adjective word, medullary region of the medulla. Now, if you watch up here, where your box is where you are in that tissue. So if I want to be down here in that cortical region, I can grab this box and come down to the cortical region. And already you can see a bunch of glomeruli. There's one, there's one, there's one, there's one. They're these little like ball-like structures. Okay, they're all over. Here's one, here's one, here's one. But we're more interested in those tubes, those tubules. I'm going to take my box and I want my box to be more in this medullary, this middle region. And already you can start to see tons and tons and tons of these tubules. Now the instructions say zoom in on a collecting duct. It could also be a tubule. I don't expect you to be able to distinguish between them. But I want you to find an area that also has blood vessels and capillaries. Let's go back to our McGraw-Hill image. In our McGraw-Hill image, let's go down to the next one, 20.11. And in 20.11, we can see blood vessels. So here's a blood vessel right here. Make this a little bigger. Okay, here's a blood vessel. There's glomerulus. There's some tubules. Okay, and over here, we can see some really tiny blood vessels next to our collecting ducts that are like little blood vessels that we're going to call capillaries. So first, stick with your low magnification. Try to find an area that has this dense, dark red on it. This is where you're going to find some larger blood vessels. I'm going to click this area. I'm going to drag it to the middle of my view. Because remember, if we don't put what we're interested in looking at in the middle, when we go to a higher magnification, we're going to lose it. Okay, it's going to be magnified outside of our view of focus. I'm going to click and zoom in here. I think I lucked out on a really good area. I like this a lot. Okay. It's actually really, really nice. So I have lots of these little collecting ducts, and all these collecting ducts are made out of our simple cuboidal epithelium. Here we've got our blood vessels, and our blood vessels are full of our blood. And if you look around the collecting ducts, you should be able to find little tiny blood vessels, little capillaries, like here and here and here. And we want to be able to have an image. There's one here, there's a little one there. We want to have an image where we have blood vessels and then our collecting ducts and then some little tiny capillaries. So we can label all of these things. Try to get it zoomed in as much as you can where you can still see all three of these structures. Okay, so here I've got my larger vessels. Here I've got my collecting ducts. Here I've got a little capillary. Okay, take a picture using your screen capturing tool. So I'm going to click on new, and I'm going to grab a picture of just this part right here. Okay, and I'm going to save that. Let's save that as cuboidal. Now I can click on choose image and select my cuboidal tissue. And I'm going to click on annotate. And that's going to bring up this annotation menu. So we want to label our simple cuboidal epithelial cells. So I'm going to draw arrows to a couple of them. I think I'll do blue. There's a cuboidal. There's a cuboidal. There's a cuboidal. Let's do one more. Why not? Okay, so we've got our simple cuboidal epithelium. We can see they're only one cell layer thick. And we've got a very, very round nuclei. I'm going to come to my text tool, and I'm going to label this simple cuboidal epithelium. Now, if you remember from the pre-lab recording, epithelial tissues are always, always, always supported by a layer of connective tissue. And between the epithelial tissues and that connective tissue, you typically have a little layer of acellular tissue that's made up of those secreted proteins and carbohydrates like proteoglycans that make up the basement membrane. Actually see the basement membrane pretty decently in this image. See if I can well, that's not going to let me point, but I can point with my giant pink arrow. So where you've got this kind of thickened line right here, or this thickened line over here, that's just outside your simple cuboidal epithelium. That's the basement membrane. So I want you to point that out. And I click on my arrow. I'm just going to change colors. Let's do yellow. And I'm going to point out my basement membrane here. Come back to my text tool. Okay. Basement. I'm going to make a new text box, say membrane. Okay. So the collecting duct is kind of like the center of the collecting tube. So that's just like the center portion. So I just want you to point that out. That's like the little channel that the fluid is going to run through as it's leaving the kidney. So what color should we do? Let's do purple. Okay, I'm just going to say that this right here is my collecting duct. Ugh, purple's not showing up too well. Let's change that to black. Okay, capillary, we're going to point out a capillary. That's a really small vessel. I think I'll circle that. Let's circle it in white. Okay, there's my capillary. And we want to point out the blood vessel. I put my labels over the blood vessels. So let me move those a bit. And now go back to my annotation tool. Kind of running low on color options here. Let's do green. Okay, I'm going to circle this large blood vessel and I'm going to label it blood vessel. Okay, so now we've got our simple cuboidal epithelial cells on the margins of our collecting duct. We pointed out the basement membrane. That's that acellular tissue between the epithelium and the connective tissue. We pointed out a small capillary, and we pointed out a larger blood vessel. So we're done with this part. I'm going to stop this video here in case anyone needs to take a break, and I will make sure everything is recorded correctly. And then for the last two slides, I will start a new recording.